Let's get into episode 12 of our cell culture series. Viability assays. Are your cells actually alive? In this episode, we'll learn how scientists measure cell health, distinguish live cells from dead ones, and ensure experimental results are reliable before moving forward. A flask full of dead cells looks almost identical to a flask full of living ones. Under the naked eye, under bright field microscope, after adding your drug, they look fine. But the data tells a completely different story. Today, how to measure cell life, death, and everything in between. The problem with trusting your eyes. Here is a nightmare scenario. You add a potential cancer drug to your cells, incubate for 72 hours, and look at the flask. The cells look fine. Confluency looks similar to the control. You conclude the drug has no effect. You publish. Later, someone repeats your experiment with an actual viability assay. 80% of your cells were dead. They just had not detached yet. Why this matters in drug discovery. Every drug screened in pharmaceutical research goes through viability assays, typically at multiple concentrations across an 8 to 12-point dose-response curve. The IC50 value generated from this curve determines whether a compound becomes a lead candidate or gets eliminated. Getting the viability measurement wrong means getting the drug discovery decision wrong. Now, let's test your understanding with a quick question. Here's the question. Why does bright field microscopy alone underestimate cell death, especially during the first 24 to 48 hours after a toxic treatment? A. Bright field cannot penetrate the flask plastic to image cells clearly. B. Dead cells retain their shape and remain attached for several hours after death. They detach later, making early cell death invisible when judging only by confluency. C. Bright field overestimates cell death because dead cells appear darker. D. Bright field is only inaccurate at high confluency. The correct answer is B. Here's why. During apoptosis, cells don't disappear immediately. They maintain their membrane integrity, stay attached to the culture surface, and often look completely normal under a bright field microscope. They only detach during the later stages of apoptosis or necrosis. That's why relying on cell appearance or confluency alone can be misleading. The key takeaway: a healthy-looking culture doesn't always mean healthy cells. Always confirm cell health with a proper viability assay. The Viability Toolkit: 8 methods, 2 principles. All viability assays fall into 2 categories. Exclusion assays: dead cells take up dye that live cells exclude, and metabolic assays: living cells convert a substrate that dead cells cannot. Each has specific applications, advantages, and blind spots. Now let's look at the 2 major categories of cell viability assays. The first category is exclusion dyes, which measure membrane integrity. These include Trypan Blue, Propidium Iodide, 7-AAD, and SYTOX dyes. They identify cells with damaged membranes, making them useful for distinguishing live and dead cells. However, they mainly assess membrane integrity and may not detect early metabolic cell death. The second category is metabolic assays, which measure cellular biochemical activity. Common examples include MTT, Resazurin or AlamarBlue, CellTiter-Glo, and CCK-8. These assays evaluate the metabolic activity of living cells and are widely used for drug screening, cytotoxicity studies, and high-throughput research. In simple terms, exclusion dyes tell you whether the cell membrane is intact, while metabolic assays tell you whether the cell is still alive and functioning. Choosing the right assay depends on your experimental goal. The critical distinction: viability versus proliferation. A cytostatic drug, one that stops cell division without killing cells, gives a flat dose-response curve in a viability assay. Not because the drug has no effect, but because the assay measures live versus dead, not growing versus stopped. To detect cytostasis, you need proliferation assays, BRDU incorporation, ADU staining, Ki67 immunostaining, or CFSE dilution. The IC50 concept. IC50 equals the concentration of drug that kills or inhibits 50% of the cell population. Calculated from a dose-response curve, plot drug concentration, log scale, x-axis, versus cell viability, percent, y-axis. The sigmoidal curve crosses 50% at the IC50. Every cancer drug in clinical development has an IC50 value. Calculating one correctly requires at least 6 to 8 concentration points, correct controls, and the right viability assay for your model system. Now, let's check your understanding with another question. Here's the question. You treat cancer cells with a new compound and find no change in the MTT signal compared to the control. Your supervisor says the compound is inactive. What alternative explanation should you consider? A. MTT is always correct. If there is no signal change, the drug has no effect. B. The compound may be cytostatic, meaning it stops cells from dividing without killing them. The cells remain metabolically active, so the MTT signal appears unchanged. C. The cells must be resistant to all compounds. DMTT only works for adherent cells, so the results are invalid. The correct answer is B. Here's why. MTT measures the metabolic activity of living cells, not whether the cells are actively dividing. A cytostatic drug can halt cell proliferation while the cells remain alive and metabolically active, producing a normal MTT signal. The key takeaway: a normal MTT result doesn't always mean a drug is ineffective. To distinguish growth inhibition from true cell death, combine viability assays with proliferation assays such as BRDU, Ki67, or cell counting over time. Running an MTT dose-response curve. The MTT assay is the foundation of drug cytotoxicity screening. Here is the complete protocol for generating an IC50 dose-response curve. Day 0. Seed cells. Seed at a density that gives approximately 50 to 70% confluency at time of treatment, typically 24 hours after seeding. Use the same number of cells per well across all wells. Day 1. Drug treatment. Prepare drug at 8 to 12 concentrations spanning 3 to 4 log units, e.g., 0.001, 0.01, 0.1, 1, 10, 100 micro-m. Include A. Vehicle control, same DMSO concentration as highest drug well. B. Untreated control. C. Blank wells, no cells. Add drug in a volume of 10 to 20% of total well volume. Incubate 48 to 72 hours. Day 3 to 4, MTT addition. Add MTT reagent, 5 mg per milliliter stock in PBS. Add 1/10 volume to wells. Incubate 2 to 4 hours at 37 degrees Celsius. Live cells convert yellow MTT to purple formazine crystals. Formazine dissolution. Remove media carefully. Add DMSO to dissolve formazine crystals. Mix well on plate shaker for 5 to 10 minutes. Read plate. Absorbance at 570 nanometers, reference wavelength 690 nanometers. Subtract blank wells. Calculate viability. Percent viability equals OD treated, OD untreated control, x 100. Plot dose-response curve. Fit sigmoidal curve to calculate IC50. The DMSO warning in drug screening. DMSO is used to dissolve hydrophobic drugs before adding to cells. But DMSO is itself toxic and can affect MTT signal at concentrations above 0.5 to 1%. Always use the same DMSO concentration in every well, including your control. A typical rule: keep DMSO at or below 0.1% final concentration in all wells. Now, let's test your understanding with a lab calculation question. Here's the question. Your MTT plate shows that the blank wells, containing only media, have an OD of 0.15 at 570 nanometers, while your untreated control wells have an OD of 0.8. If you forget to subtract the blank, how does your calculated IC50 change? A. IC50 is unaffected because blank wells contain no DMSO. B. IC50 is overestimated because the inflated baseline makes treated wells appear more viable than they actually are, shifting the dose-response curve upward. C. IC50 is underestimated. D. The effect depends on the cell line being used. The correct answer is B. Here's why. The blank wells contain background absorbance from the media, plastic plate, and assay reagents. If you don't subtract this background, every absorbance value becomes artificially higher. As a result, treated cells appear more viable than they truly are, causing the dose-response curve to shift upward and the IC50 value to shift to a higher concentration. This makes the drug appear less potent than it actually is. The key takeaway: always subtract the blank wells before calculating cell viability or IC50. It's an essential step for obtaining accurate and reliable results. 4 mistakes in drug screening and how they affect IC50. Mistake 1. MTT in empty wells. The fix: Always include blank wells, media, MTT, DMSO, no cells, in every plate. Subtract blank OD from all readings before calculating viability. Without this correction, background contributes to every viability value. Mistake 2. Unequal cell density across wells. The fix. Seed cells with a multi-channel pipette or electronic multi-channel pipette for consistency. Mix cell suspension well before each row. Edge wells often receive slightly different volumes due to pipetting angle. Use edge wells only as blanks are controls, not treatment wells, if edge effects are problematic. Mistake 3. DMSO concentration inconsistency. The fix. Calculate the final DMSO concentration in every well, including the vehicle control and the highest drug concentration. Make them equal. Many drug stock solutions are in 100% DMSO. Serial dilutions in media still leave significant DMSO in high concentration wells if not equalized. Mistake 4. Confusing viability with proliferation. The fix. If your compound is expected to be cytostatic, e.g., cell cycle inhibitors, kinase inhibitors, run both viability and proliferation assays. Report both. A compound that appears inactive by MTT may be profoundly cytostatic. Missing this leads to false negatives in drug discovery. Episode 12 Summary. What you now know. 1. Exclusion dyes measure membrane integrity. Metabolic assays measure biochemical activity. Trypan Blue and Pi test membrane permeability. Are cells intact? MTT and CellTiter-Glo test metabolic activity. Are cells biochemically alive? Both measure viability but capture different aspects of the death process at different times. Use the right tool for the question and the time point. 2. MTT is the gold standard for drug screening cytotoxicity. Cheap, robust, widely validated, 40+ years of use. The 96 well, 384 well, and 1536 well formats scale from academic lab to pharmaceutical HTS automation. Always include proper controls. Blank, vehicle, positive control, known toxic compound. Always subtract blank, always normalize to untreated control. 3. Always distinguish viability, alive, from proliferation, growing. A cytostatic drug looks inactive by MTT. A cytotoxic drug looks active. Many important drug classes, cell cycle inhibitors, differentiation inducers, cytostatic natural products, show their primary effect in proliferation assays, not viability assays. Always ask, could this be cytostatic? Then add the proliferation measurement. Thank you for watching the episode 12 of Cell Culture series. In the next episode we'll dive deeper into 3D culture and organoids. See you there. If you found this episode useful, don't forget to subscribe our channel. Don't forget to like, share and leave the comment below. We'll see you in the next episode. Thank you for watching.